Hello everyone, this is Murari Monohar Nath from 4th semester, Department of Zoology, North Lakhimpur College. Today, I am going to present my departmental seminar under the guidance of Bipul Soikia sir, and the topic of my seminar is Life History and Pathogenicity of Pork Tapeworm. So, here are the content. Now, moving on to the introduction. Tapeworm are ribbon-like creatures which are found as endoparasites of man. They are helminth worm, which belongs to the phylum Platyhelminth. They are responsible for causing the disease Taeniasis, and they are digenetic parasites. Here on the left, we can see a picture of a tapeworm inside the intestine of man, and on the right, we can see a diagrammatic representation of a tapeworm showing its hook, rostrum, scolex, sucker, neck, and proglotid. There are mainly three types of tapeworm which infect human. They are pork tapeworm, which is Taenia Solium, beef tapeworm, Taenia saganita, and fish tapeworm, Diphyllobothrium latam. Here, we can see the pictures of pork tapeworm, beef tapeworm, and fish tapeworm respectively. Now, moving on to the systematic position of pork tapeworm. Pork tapeworm belongs to phylum Platyhelminth, class Cystoda, subclass Eucystoda, order Taenioidea or Cyclophilioidea, genus Taenia, and species Solium. Next is distribution. Pork tapeworm is cosmopolitan in distribution. It is found in India, America, Africa, few European countries, and Australia. It is rare in Middle East and Muslim majority countries, as consumption of pork is banned among the Muslim community. Here in this picture, we can see the distribution of pork tapeworm. Next is the structure of tapeworm. Pork tapeworm's body is ribbon-like, dorsoventral flattened and elongated. Its body is differentiated into three regions, which is head or scolex, neck, and strobila. The following picture shows us the diagrammatic representation of a tapeworm showing its head, neck, and strobila. The strobila contains about 700 to 900 proglotid. Next is head or scolex. Head is a knob-like structure which contains rostrum and suckers. The rostrum contains 30 to 40 chitinous hooks which are arranged in two rows. The head is only for attachment and not for feeding. Next is the neck. Neck lies behind the head or scolex. It is a short, unsegmented tube-like region. It is also called zone of proliferation or zone of overgrowth, as proglotid arises from the neck. Here on the right, we can see a picture showing us the head and neck of a tapeworm. Next, moving on to strobila. The rest of the body of the tapeworm is called strobila. The strobila contains about 800 to 900 proglotid, and each proglotid contains about 300 to 400 tapeworm eggs. Next is life cycle of pork tapeworm. Taenia Solium has a digenetic life cycle, which means they requires two hosts, one primary and another secondary, to complete its life cycle. The primary host of Taenia Solium is man, and the secondary host of Taenia Solium is pig. Now let's see the life cycle of tapeworm in primary host. So the life cycle of tapeworm in primary host starts with copulation and fertilization. Copulation takes place when the series, which is the male copulatory organ of tapeworm, is inserted into the vagina of the gonophore of same or different proglotid. Generally, self-fertilization takes place as there is only one tapeworm present in one host. Next is formation of capsule. After fertilization, the eggs become associated with yolk cell in the ootype. Both egg and yolk become enclosed in a thin capsule. The capsule is formed from the materials of the yolk cell. Here in the left picture, we can see the transformation of a fertilized egg into morula and oncosphere. And on the right, we can see a picture showing us the egg of a tapeworm. Next is formation of oncosphere. The development of the capsule takes place in the uterus, where the zygote undergoes holoblastic unequal cleavage to form the morula. The morula develops into the hexagrant embryo, which has 6 hooks and a pair of penetrating glands. The hexagrant embryo with the capsule is called oncosphere. On the extreme left, we can see the oncosphere without shell and with shell. And in the middle, we can see the hexagrant embryo. And on the right, we can see the egg of a tapeworm. Next is formation of gravid proglotid. By the time oncosphere is formed, the proglotid becomes large and gravid, and the gravid proglotids get detached from the strobila and is released to the outside with the faces of host. Each gravid proglotid contains about 7 to 13 branched uterus, and each proglotid contains about 30,000 to 40,000 eggs. Next is, here we can see the picture of a proglotid along with its branched uterus, and on the right we can see the reproductive organs of a tapeworm. This is a diagrammatic representation of the life cycle of Taenia Solium, showing us both the primary host and secondary host. Now, life cycle in secondary host. Infection to secondary host. The secondary host of Taenia Solium is pig, which acquires the infection by ingesting oncosphere when they feed on contaminated human excreta. Next is migration of oncosphere within the host. When the oncosphere reaches the stomach of pig, it loses its basement membrane due to the action of stomach acid, and the oncosphere gets transformed into the hexagrant embryo. With the help of hooks and penetrating gland, the hexagrant embryo penetrates the intestinal wall and travels to the liver through the submucosal blood vessel. From liver, it travels to the heart and enters the arteries. Through the arteries, it travels to the muscles. The hexagrant embryo in the muscles gets transformed into bladder worm by absorbing nutrients from the host tissue, and the bladder worm later gets transformed into cyst. These cysts are not only found in muscle but also found in brain and lungs and liver of the host. Here, the left picture shows as a oncosphere, the middle picture shows as a hexagrant embryo, and the right picture shows as a bladder worm. Next is infection to primary host. When a man eats undercooked food, bladder worm reaches the intestine and gets activated. The bladder worm then attaches itself to the intestinal wall with the help of hooks and suckers. In about 10 to 12 weeks, the bladder worm develops into adult tapeworm. Here, these are the picture of pork meat which contains tapeworm cyst. When a person eats such meat, the person acquires the infection of tapeworm. Here, we can see the different stages of tapeworm from fertilized egg to bladder worm. Now, moving on to pathogenicity of tapeworm. Taenia Solium is responsible for causing teniasis and cysticercosis in man. So now, let's move on to teniasis. Teniasis is caused by adult tapeworm when it infects the intestine of man. Here, I have attested a video which shows tapeworm in the intestine of man.
Okay, I think we can see some much bigger segments here. This is further down the length of that worm, getting towards the back end of it. The segments get bigger, they get more mature. Here we've got the biggest segments here. So these segments are probably about a centimeter or so in size.
So now, moving on to the symptoms. The symptoms of teniasis are abdominal pain, nausea, anemia, indigestion,
Okay.
weight loss, and internal bleeding. Teniasis can be diagnosed by examining the patient stool for eggs of tapeworm and by testing antibodies in blood for tapeworm. So now treatment. Teniasis can be treated by using antihelminth drugs such as hetrozan and mepacrine. It can also be treated by removing the tapeworm cyst by surgery. Next is cysticercosis. When the infection is caused by larval stage of tapeworm, it is called cysticercosis. When the cyst of or larval tapeworm occurs in brain and the central nervous system, it is called neurocysticercosis. So now, let's see the symptoms of cysticercosis. The symptoms of cysticercosis are abdominal pain, nausea, swelling of eye and low vision when the cysts occur in eye, formation of lump in various parts of the body and under skin, seizures and headache when the cyst occurs in brain and central nervous system. So diagnosis. Cysticercosis can be diagnosed by examining patient stool for eggs, by testing for antibodies for Taenia Solium, MRI scans, CT scans of brain and X-ray scans. Here, in the left picture, we can see a CT scan of brain. Here the white spots in the brain are tapeworm cyst. The middle photo is a X-ray of cyst. Here the white spots are cyst of tapeworm. So now treatment. Cysticercosis can be treated by using antihelminth drugs such as hetrozan and mepacrine and removing the cyst by surgery. Here in the photo, we can see a tapeworm being removed from the brain of a patient by surgery. Moving on to prevention of teniasis and cysticercosis. Teniasis and cysticercosis can be prevented by taking up the following measurement, which includes: number 1, washing hands with soap. Number 2, washing fruits and vegetables before eating. Number 3, proper disposal of human and animal excreta. Number 4, cooking meat up to 150 degrees Fahrenheit or 65 degrees Celsius. Number 5, freezing meat and fish up to 7 days, which will kill the tapeworm eggs and larvae. And number 6, not consuming raw meat or undercooked beef, pork and fish. Now, moving on to conclusion. From the above seminar, I would like to conclude that pork tapeworm is an digenetic endoparasite which lives in the intestine of man and muscles of pig. It is responsible for causing diseases such as teniasis, cysticercosis and neurocysticercosis. It is an obligate parasite which cannot live without an host. It can live up to 30 years and can grow up to 10 meters in length. Tapeworm infection can be treated by antiparasitic medication and by surgery. Personal hygiene and proper cooking of food is the key to avoid tapeworm infection. So, here are the references, the websites and the books which I took the reference from. The website includes Google, Wikipedia, Study and Score, and the books include Modern Textbook of Zoology by R.L. Koptal, Parasitology, and NCERT Class 12 Biology. Thank you and have a beautiful day ahead.